My name is Dan Sohm. I'm Principal Scientist and Director of Marketing at Wyatt Technology Corporation, headquartered in Santa Barbara, California. At Wyatt, we focus on applications of light scattering for basic characterization of macromolecules and nanoparticles. And so we have a product line that we present as the light scattering toolbox for essential characterization of biophysical properties, macromolecular nanoparticle properties. And that includes molar mass, size, charge, interactions, conformation, as well as composition of these various entities. And so you can see here some of the instruments that we provide. Our flagship instrument is the Dawn Helios, and we actually have here the latest version of the Dawn, which is the Micro Dawn, just launched, and this is targeted at the UHPLC market. And what the Micro Dawn does is take the basic light scattering capabilities, which we use downstream of HPLC, size-exclusion chromatography, to characterize the molar mass and size of macromolecules and proteins independently of elution time, and bring those capabilities over to UHPLC. So what we've done is we've shrunk the flow cell, shrunk all the internal fluidic components to reduce the dispersion, and now we have the very small dispersion that is suitable for the really narrow peaks that you get out of UHPLC. Another product that we have that uses light scattering is the Mobius. The Mobius measures charge using light scattering. We do that via electrophoretic mobility combined with dynamic light scattering. So the electrophoretic mobility gives us the electrical properties. We combine that with dynamic light scattering, which gives us a measure of the size of the particle. And when you combine that information, you can actually determine the zeta potential of the nanoparticle or the molecular charge of the protein. Third product that utilizes same principles is the Dynapro plate reader. The plate reader uses standard microwell plates, just like these, makes the measurements in situ, and what it allows you to do is characterize size and aggregation of nanoparticles, proteins, all kinds of macromolecules. And you can do it sampling hundreds of different conditions or different formulations. The measurements are made in situ in the microwell plate. And so there are no fluidics, no handling, no issues of carryover, and the measurements can be very quick. In fact, you can run through a 384 microwell plate in about an hour and a half or 2 hours and complete that entire measurements. The Dynapro also allows you to ramp the temperature. So for instance, if you want to characterize melting temperature of the proteins, it can do that, and you can simultaneously measure all these different formulations. Many other capabilities of the plate reader include characterizing colloidal stability, through second virial coefficient and the diffusion interaction parameter, aggregation, size, distributions of sizes for particles, aggregates, macromolecules, and proteins. So we have, this is just a selection of some of the instruments that we have. Some of the other capabilities include characterizing macromolecular interactions. So for instance, protein-protein binding. We do that via a technique called composition gradient light scattering. And so we have another device which is similar to a stop flow device and mixes up different compositions of proteins with each other or with dilution, injects them into the light scattering detectors. The light scattering measures the absolute molar mass of the solution. And when complexes form, the solution molar mass increases, and by teasing out that relationship with composition and concentration, you can actually determine binding affinity as well as absolute molecular stoichiometry rather than just stoichiometric ratio. And all this is done in solution without any labeling or without any immobilization. We have some other instruments, which you might see here on our posters. For instance, the Nanostar. So Nanostar is another dynamic light scattering instrument. It's a traditional cuvette-based dynamic light scattering instrument for measuring size. But one of the unique things about the Nanostar is that it also incorporates a standard static light scattering detector, optimized for static light scattering, and so allows us to measure molar mass as well as the size. And by combining information about size and molar mass, you can learn something about the conformation of the molecules. Other instruments that we provide that are not based on light scattering but work together nicely with the light scattering, also downstream of UHPLC. So we have a refractive index detector, which is unique because it has a range which is basically about 50 times larger than the range of any other online refractive index detector. We have an online viscometer, which is more sensitive basically than any of the other viscometers out there. So we basically are supplying the highest quality data so that researchers don't really waste their time with poor data. Of course, we have many capabilities in the software as well for some powerful analyses. One of the popular analyses for people working on protein conjugates or copolymers is what we call the conjugate analysis, and that allows you to determine in your eluting peaks the composition of your macromolecules, in particular when they're composed of, say, a protein linked to a carbohydrate or two different types of copolymers. So just beyond standard molar mass and size, there are many other things that we can learn about the particles and the macromolecules. If you'd like to learn more about some of the capabilities of the instruments that we provide and how they can benefit your lab, I encourage you to visit our website, www.wyatt.com. We have a host of information there, which includes introductions to light scattering, some of the theory, various application notes on how to utilize these for different types of analytes, as well as an extensive bibliography of peer-reviewed articles that cite our instruments, where the researcher has used our instruments to collect the data. And that bibliography is approaching 10,000 articles at this point. So there's quite a lot that you can learn. I encourage you to contact us as well as to check out the website. Thanks.